In this video, we're going to be talking about HPV and how it leads to cervical cancer, and kind of use this as a prototype for viral-induced cancers in general. And so HPV stands for human papilloma virus, and infection with HPV can lead to benign papillomas, which is another word for warts, and any number of various genital cancers, as well as cancers of the oropharynx or the throat. But HPV is probably most associated with cancers of the cervix, since nearly 100% of cervical cancer is linked to HPV. And there are over 30 to 40 types that can be transmitted by sexual contact. And some specific types to be aware of are types 6 and 11, which cause warts, and types 16 and 18, which are the most carcinogenic and thus responsible for most cases of cervical cancer. Alright, so HPV has two viral oncogenes in its genome called E6 and E7. And these incorporate into host DNA and are transcribed into proteins that inactivate host tumor suppressor proteins. So E6 inactivates P53. And it does this via ubiquitin ligase activity, which acts to ubiquinate P53. And this leads to P53 degradation. Now E7 inactivates PRB, and it does this by competing for the retinoblastoma protein binding site. And this ends up freeing transcription factor E2F to activate its targets, which in turn pushes the cell cycle forward and leads to increased cell growth and cell division. Once an HPV infection takes root, how does it progress from infection to cervical cancer? Well, the earliest changes to cervical epithelium resulting in cervical dysplasia are visible by biopsy. And these changes have a special term, and the term is called cervical intraepithelial neoplasia, or CIN for short. And there are actually 3 grades of CIN, but first let me bring up a slide of some normal tissue so you can see what that looks like. Alright, so this is a slice through some normal cervical epithelium. Here is the outer surface, and here is the basement membrane. And so overall it looks organized, and the cells are relatively similar shape and size with normal sized nuclei. So now let's bring up the first of the 3 grades, CIN1, and see what that looks like. Alright, so here is an example of what CIN1 looks like. So now you can see there's some mild dysplasia, and it's confined to the basal 1/3 of the epithelium for the most part. And so this usually corresponds to an infection with HPV and can be cleared by the immune response, but sometimes it takes several years for this to clear and return to normal. Now the next grade is CIN2, and you can see there's some moderate dysplasia now, and now it's confined to the basal 2/3 of the epithelium. You also start to see some sort of increased nuclear to cytoplasm ratios, as well as some mitotic figures. And the last grade is CIN3, which I'll bring up here. And so here you can see there's severe dysplasia now, and it spans more than 2/3 of the epithelium. And in the cases where it involves the full thickness of the epithelial layer, we call this carcinoma in situ. And another thing to be aware of is that CIN1 is considered to also be a low-grade squamous cell intraepithelial lesion, or LCIL, whereas CIN2 and CIN3 are considered to be high-grade squamous cell intraepithelial lesions, or HCIL. So that's sort of a broad overview of cervical cancer and HPV as sort of a prototype for viral-induced cancer. And there are also several other well-known viral-induced cancers. And let's make a little bit more room here to write those out. And some examples of those would be Burkitt's lymphoma, which is due to Epstein-Barr virus, or EBV. There's hepatocellular carcinoma, or cancer of the liver, which I'll abbreviate as HCC. And this is linked to hepatitis B as well as hepatitis C virus. There's also adult T-cell lymphoma, which is linked to human T lymphotropic virus 1, or HTLV1. And the last example that I'll give you is Kaposi sarcoma, which is linked to human herpes virus 8, or HHV8.